Yeast Two-Hybrid system. Do you know that proteins in our cells often work together? They need to interact with each other to complete a specific task in the cell. If these proteins fail to interact, the task remains incomplete. The Yeast Two-Hybrid system is basically a lab trick. It helps us discover whether two proteins interact with each other or not. To figure it out, we use a transcription factor called GAL4. It is a protein that can transcribe genes. The GAL4 protein has two units, activation domain AD and DNA binding domain BD. Normally, these two domains stick together and turn on or transcribe a reporter gene. When they get separated, the GAL4 protein fails to turn on the reporter gene. We intentionally separate these two units to find out whether two proteins, protein A and protein B, interact or not. If protein A and B interact, those two units of the GAL4 protein get pulled back together. That reunited protein can now turn on the reporter gene. If the proteins A and B do not interact, the two units of GAL4 stay apart and the reporter gene stays off. In Yeast Two-Hybrid system, we use two separate plasmids. The first plasmid has two genes, one for the DNA binding domain and the other for protein A. The second plasmid also contains two genes, one for making the activation domain and the other one for protein B. The goal here is to find out whether proteins A and B interact with each other or not. So we transfer both plasmids into a yeast cell. Inside the cell, plasmid 1 produces protein A bound to DNA binding domain and the second plasmid produces protein B fused to the activation domain. If protein A and B interact, both units of GAL4 protein reunite and transcribe the reporter gene that produces nutrients. As a result, the yeast cell grows on the plate. If both proteins do not interact, the GAL4 units stay apart. The reporter gene does not produce nutrients. As a result, the yeast cell does not grow